Hello, I'm Dr. Trish Murray, physician, best-selling author, and the Health Catalyst speaker. And you found this talk most likely because you've been searching to try and understand better how the COVID-19 vaccines work. And that's exactly what I'm going to cover here. So let's get going. So first of all, to understand the mechanism of how the vaccines work, we have to understand what is a virus. So how would you attack it? So viruses are microscopic parasites, generally much smaller than bacteria. Viruses are made up of a small collection of genetic code, could be DNA or RNA, and that genetic code is surrounded by a protein coat. Viruses cannot replicate alone out in the environment. They must infect cells. So they must infect an animal or a human and get into your cells. And then they hijack the host cell's machinery, basically your genetics, in order to replicate. Typically, they then kill the host cell in the process of replication and cause damage to the host organism. Researchers have estimated that viruses outnumber bacteria by more than 10 to 1, and viruses cannot be killed by antibiotics. That's why we've been in a pandemic for the last year, because antibiotics are of no use against a virus. Only antiviral medications, but we don't have any specific antiviral medications for COVID-19. Or then, in order to protect or get your immune system to be able to protect you, vaccines can eliminate or reduce the severity of viral disease. Now let's look at the definition of a vaccine. A vaccine is a product that stimulates your immune system to produce immunity to a specific disease, therefore protecting you or any person from that disease. Now again, with what we just said, if your immune system is ultimately what's going to protect you, then you need to have some basic understanding of how the immune system works. Now the immune system is a very complex system, but I'm going to take a few moments here and give you the basics so that you have a basic understanding. So first of all, you've got to understand that your immune system is a defense system. It is your military with different branches. Just like in our American military, we have the Navy, the Army, the Air Force, the Marines. There are different branches to your defense systems of your body in your immune system. So those branches in your immune system are called the innate immune system, the adaptive immune system, and the adaptive immune system, again, can have different branches, such as the Th1 pathway and the Th2 pathway. Now, I'm not going to go into huge, complex detail about all of these different branches. I'm giving you the basics. But I'm also going to later give you an offer of my first book for free, in which I talk a lot more about the immune system and how it works. But let's talk about the different branches. So the innate immune system is really your ground forces. It's your army, the men on the ground, the men and women on the ground forces that are the first line of defense against any invader. So your innate immune system is the first thing to attack any virus or any bacteria or any foreign invader of your body. The adaptive immune system is trained by the innate immune system over the first 4 or 5 days of any either bacterial or viral infection. And then what happens is they are the reinforcements. The adaptive immune system are your reinforcements, your other branches that become trained in what to attack and how to identify them. And they come into play as a second form of attack against any invader. And it is the adaptive immune cells that can be trained into one of 2 different branches of your immune system, the Th1 or the Th2 pathways. And it is this immune system, these branches, the adaptive immune cells, that create antibodies or even something called T cells that then have long-lasting, long-term memory against any invader. So if you see them again, they don't need to be retrained. They've already been trained and they can go into action right away. Now, again, this is my first book, Make a Dent in Chronic Disease. And if you stay to the end, you're going to realize that I'm going to give you this book for free for listening to this presentation. And in this book, I give you much more detail about the immune system and what you can do to optimize your immune system in preparation for the vaccine, or obviously to protect you optimally against any viral or bacterial infection, and particularly against COVID-19. So now let's start looking at the mechanism of the different COVID-19 vaccines. And again, before we get into each different mechanism, let's look at the coronavirus itself. Now, on the left side of this slide, you see a diagram of the basic diagram of the coronavirus. You see the spike proteins on the membrane of the virus, and you see the virus genome inside the virus. So again, the spike proteins on a coronavirus have a crown-like appearance or shape, and that's how corona got its name. So the primary target in all of the vaccines against coronavirus or COVID-19 is the S or the spike protein that's on the membrane of this virus. So let's start going through the three different mechanisms that are presently being used in order to create vaccines against the COVID-19 coronavirus. Number one is called the purified protein with adjuvant vaccine. And this is the most classic type of vaccine because it's been used in history numerous times. It's presently in use against hepatitis B, against tetanus, and against diphtheria for many years. What they've done is the spike protein is isolated. The protein itself is isolated from the COVID-19 coronavirus and then replicated. And that isolated S protein or spike protein that now can be replicated is injected into the body of the host or anyone trying to initiate an immune response. And that stimulates a weak immune system response usually. So that's where the adjuvant comes in. An adjuvant is a chemical stimulant that irritates your immune system even more and boosts your immune system response into a more robust response. The spike protein, once injected into you, is recognized by your immune system as foreign and then initiates the ground forces, the innate immune system, to attack. And then the innate immune system takes messages along with them and starts training your adaptive immune system to bring in reinforcements within 4 or 5 days and creating antibodies against the spike protein to develop a lasting long-term memory against the spike protein. Now, folks, there are different companies like GlaxoSmithKline or Novavax that are still in trials. These vaccines, this type of mechanism, the purified protein with adjuvant, the classic vaccine takes a long time to develop and get through trials. So this type of vaccine is not in use or available yet. Here is a chart to just simply simplify and review what we just went over with protein vaccines. And this is by the British Society of Immunology. So all the way to the left, you see the coronavirus diagram, and they take those spike proteins off and they replicate. So protein vaccines contain proteins from the COVID-19 virus. So the SARS-CoV-2 is the corona COVID-19 virus, which are recognized by your immune system to trigger a response. These could be whole proteins, protein fragments, or they could even take many protein molecules and pack them in nanoparticles. That's what's injected, and this response builds immune memory so your body can fight off the SARS or COVID-19 virus in the future. Again, over here to the right, these have good previous safety records. They're usually administered again with an adjuvant to boost immune response. Examples that have been used throughout history are hepatitis B vaccine. And again, presently in clinical trials for COVID-19 are a couple different companies' vaccines, but they are not in use yet. The second mechanism that's available and is in use so far is called the viral vector vaccine, or the carrier vaccine. An example that's been used in history has been a vaccine against smallpox. Now, what do they do for this? This is, they go in and they literally decode the DNA genetic sequence for the production of the spike protein, meaning they go into the coronavirus, they take out the genetics of it, they study it, and they can replicate the DNA of the virus, and they insert the DNA that encodes specifically for the coronavirus spike protein in another virus that's less dangerous. And the virus that's being used is the adenovirus. The adenovirus typically causes the common cold or bronchitis. And so they replace the adenovirus's genetic material with the specific DNA that encodes for the coronavirus spike protein. Once they've done that and genetically modified the adenovirus, they then inject you with that adenovirus. And after vaccination, the modified adenovirus infects your cells. It does what viruses do. It attaches to your cells, it gets inside your cells, and it inserts its DNA instructions for the spike protein into your DNA. And it is your cell's DNA that then creates what's called messenger RNA. This is the machinery. Your DNA creates messenger RNA, and messenger RNA then is the code that produces proteins. So your cell's DNA then create messenger RNA, then in turn signals the production of spike proteins and places them on your cell membranes. And of course, your immune system is activated by the invasion, if you will, or the foreignness of now your cells with spike proteins on them. And your innate immune system attacks those cells that have the spike protein, and then it kills them, and then goes and trains the adaptive immune system to come in as reinforcements. And as a result, your adaptive immune system develops antibodies and T cells that have memory against the spike protein for any future infection. Now, the example that's being used here in the United States presently is the Johnson & Johnson vaccine. AstraZeneca, that was produced in the United Kingdom, is presently trying to get approval in the United States, but it's been used in the UK and in parts of Europe already. Sputnik is a viral vaccine, a viral vector vaccine that was developed and is being used in Russia. So let's summarize again the steps we just did for the viral vector vaccine. So all the way to the top left corner, you see a diagram of the coronavirus. And you see that the genetic material is being removed and it's being put into a different virus, the adenovirus. So you use an unrelated harmless virus, and that virus is modified to deliver the COVID-19 genetic material. The delivery virus is known as a viral vector. And that is what is injected into you. And your cells are then infected by the adenovirus, and the genetics of the adenovirus are put into your genes, into your DNA. And it's your cells and your genetic material now that's infected with the right genetic material, make the specific protein, the spike protein, and puts it on your own cells, which then are recognized by your immune system to trigger a response. And this response builds immune memory, so your body can fight off the COVID-19 or coronavirus in the future. Under considerations on the right here, many times the viral vector vaccines generate a very strong immune response. But many times, in order to keep them stable, they need to be stored at specific low temperatures before injected. Again, AstraZeneca, when they created by the British Society of Immunology, is what was being used as the example here, but Johnson & Johnson is also a viral vector vaccine. The Ebola virus that was used in Africa, the Ebola vaccine used in Africa is also a viral vector vaccine. And there are other companies that still are in clinical trials with other viral vector vaccines. Let's go through the third mechanism of possible use, and that is already in use for COVID, which is the messenger RNA vaccines. Now again, messenger RNA is a form of information produced by DNA that cells throughout the body use to produce what? Produce proteins. So science broke down the spike protein from coronavirus, and they analyzed it to determine the specific messenger RNA sequence that encodes for the spike protein. Scientists then replicated the messenger RNA sequence. They then took this messenger RNA code sequence and they packaged it. You see, messenger RNA is very fragile. It can be broken down easily by heat or by the enzymes in your body. So they need to protect it. So they package the messenger RNA coding sequence in a lipid. Lipid is fat, in a fat particle. So a lipid nanoparticle. Nano just means really, 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 really small. So a lipid nanoparticle in order to protect the messenger RNA. And it's the lipid nanoparticle with the messenger RNA coding inside it that's injected into you. And after the injection, the protective lipid covering, meaning again, the fat coating that's surrounding the messenger RNA binds to the membranes of your cells. Our cells are made of lipid fat. So the fat likes fat, so they go and get absorbed. So the lipid coating gets absorbed, attached, and then absorbed into through your cell membranes. And then the messenger RNA code is therefore emptied into your cell cytoplasm. And that messenger RNA code is already ready to start processing protein. So the messenger RNA code then messages for the creation of a spike protein. That is then, your body then puts those proteins where they are told to go, which is on the membranes of your own cells. And as a result of your cells having spike protein on them, your immune system sees them as foreign and attacks. So it initiates an immune system defense. The messenger RNA, though, does not last long, and it breaks down in cells as it is very fragile to your body's enzymes and to the heat, of course, of your body. So that's why, and I'll talk about in a minute, we need 2 shots. So examples of this vaccine have been in use here in the United States, the quickest, and that's been Moderna and Pfizer, and they are both messenger RNA vaccines. So let's review these steps one more time. And again, these are charts from the British Society of Immunology. And these are considered, they've been called a category of, quote, genetic vaccines, nucleic acid vaccines. So up in the top left corner, you see, again, the diagram of the coronavirus. And you notice that they took a piece of the genetics, and then they took the messenger RNA, and they figured out the code for the messenger RNA. So what happens is you contain a segment of the coronavirus genetic material that codes for a specific protein. Now here it says this can be DNA or it can be RNA, but as far as I understand it, these are messenger RNA vaccines. And our cells use this messenger RNA, this genetic material, to make the COVID-19 protein, the S protein, the spike protein, which is recognized by our immune system to trigger response. And of course, this response builds immune memory so your body can fight off the COVID-19 infection in the future. Over to the right, considerations. These are low cost and fast to develop. These are new. They've not been used in the past. And again, in order to protect the messenger RNA, they may need to be stored at very low temperatures because they're fragile. Examples in use are Pfizer and Moderna, and the Imperial College of London does still have one of these vaccines in clinical trials. Now, why are 2 shots necessary with the messenger RNA vaccines? Again, the messenger RNA vaccine the messenger RNA itself is fragile. So the immune system response to the first messenger RNA vaccine is typically pretty weak and doesn't last long enough. And that's why a second shot initiates a more robust immune system response to fully train and fully engage the adaptive immune system to come on board and develop long-lasting memory against the spike protein. So let's summarize. First of all, there are 3 different vaccine mechanisms, but only 2 are available presently. All viruses, folks, are parasites that must use the machinery of any infected host to replicate. So they use your genetics. And that's why you see these different vaccines using your genetics in order to produce an immune response. So the 2 being used presently, again, are the viral vector vaccines. Johnson & Johnson, for example, or AstraZeneca, if and when it's approved here in the United States, injects you with an adenovirus whose DNA has been changed to include the correct genetic information to encode for the coronavirus spike protein using your cell's genetics. And it only requires 1 shot. The messenger RNA vaccines, Pfizer and Moderna, inject you with an already developed code for the coronavirus spike protein in the form of messenger RNA that has been synthesized scientifically. And it requires 2 shots because messenger RNA is fragile and doesn't last long enough to get a robust enough immune system response. So I'm going to stop sharing and come on. And I sure hope that this has helped you understand the mechanism of the different viral vaccine options that are out there and available to you. And I hope it helps you decide whether to proceed with a vaccine or not. Now, if you want to learn more, again, I am offering you for free my first book, Make a Dent in Chronic Disease. And if you are interested in the book, where in the book I explain your immune system much more in depth, and I give you very specific steps based on diet, exercise, nutrition, and transformation of stress in order to optimize your immune system, either in preparation for the vaccine or in order to protect your immune system and optimize your defenses against any disease process, but particularly against COVID-19. So if you want a copy of this, a free copy of my first book, just follow the link on the page connected to this video. And I hope to talk to you in relation to that some more.